be talking to you about advances we've made to using endothelial progenitor cells as a starting cell substrate for reprogramming to produce iPS cells. The work I'm describing was done predominantly whilst I was based in Cambridge, although a few little bits were done whilst I got to UEA. So let's move on. The main thing, or the first thing to talk about, is that for this audience, I'm sure you understand or know what iPS cells are. But what makes them really great, and the whole discovery of them really helpful, is that we are now able to really model human development and also, importantly, human disease. And this has been a very helpful forward step in the field. But of course, beyond modeling, what we'd also like to do is develop ways to take iPS cell technologies into cellular therapies and tissue engineering. So in this talk, what I really want to then talk about is how we can generate iPS cells in a way that we think produces higher quality iPS cells, but also a process in which we can then make these cells usable for clinical translation. So there are two papers that I'm going to talk about predominantly again in this talk from our lab. The first one from Stem Cells Translational Medicine, and the second one in New Biotechnology. If a lot of what I'm saying will be from those papers, so should you want to refer to those after my talk, you can get a lot of information. And again, we're focusing on quality and the process. So moving towards clinically compatible iPS cell generation, we need to think of several things, because on the one hand, although we want to produce these under good manufacturing processes, we also, for the sake of clinical translation, have to consider advanced therapeutic medicinal purpose as well. So that requires a higher grade, higher level of manufacture. So that means that we at least need to consider the somatic cell type we're using, the transgene delivery method, the cell culture method, and I said the quality of the iPS cells that we produce at the end of this process. Now, starting with the first, let's think about cellular therapies, for example. When we derive our cells in the first place from our individuals of choice, our patients, it turns out that skin biopsies, which are the traditional way for isolating your skin fibroblasts to make your iPS cells, are not suitable for all types of patients. If we were to look at the young, for example, or the very old, it might be undesirable to go to those. So some parents don't like the idea of you taking a skin punch from your sick child in order to produce iPS, or the skin fibroblasts to make the iPS cells. But although that's undesirable, there are other situations where it's really either highly undesirable or just not really possible. So in my field of cardiology, cardiac regenerative medicine, most of the patients we see are on blood thinners like warfarin. Now, if I was to take a blood sample, sorry, a skin sample from one of those patients, of course, they would just bleed, and that's really undesirable. But then there are other situations, for example, patients with wound healing problems, and a disease here I'm showing you on the right-hand side of my screen. It's called fibrodysplasia ossificans progressiva. And in this particular case, what you're looking at is the individual that has this disease here, and then their skeleton is actually on the furthest of the screen on the right-hand side. And this actually skeleton you can actually find in the Smithsonian Museum in the US. So this person has a disease called fibrodysplasia ossificans progressiva, or FOP for short. And what happens in this disease is that any site of trauma or inflammatory insult causes ectopic bone formation. It's a horrible disease. It starts anteriorly, where you start getting ectopic bone formation in the neck, and then in the face and the jaws, and then moves it down through the shoulders into the arms, eventually working its way down into the rib cage of the individual. And of course, as you can imagine, if you're making lots of ectopic bone that's sticking together other parts of bone, think of the rib cage, you're going to really struggle to open your chest and breathe normally after a while. And it's a slow and quite horrible death. So we would never go to a patient like this and say, can we take a skin biopsy from you? Because that would cause some large catastrophic event for them. So skin biopsies are not suitable for many patient cohorts. So we need alternative sources of cells. Efficiency is also a big issue. So in the UK, we have a system called the NHS for our health service, but around the world too, whatever health service you have, if you want the health service to start picking up the tab for making iPS cells for any individual or for all individuals, it's got to be cheap and it's got to be fast. And then the final issue that I really have to think about is genetic integrity. So this issue is something that came up after the initial iPS cell lines were generated. People started to look at the genetics of these cells. And here from a paper in 2011, what they discovered was that when you look at copy number variations using SNP arrays in individual cell lines, what you find is in the early passages, if we focus on this diagram here, on the right-hand side, sorry, you can see there's a big number of copy number variations in early passages. And although these sort of thin out over time, they're still at least in the sort of 25 to 50 range of copy number variations noted per cell line. Now, the reason this drops is partly because what's happening is that there's a predominant cell within your population of iPS cells, which has probably got the most copy number variations, or the ones which are pushing for or driving proliferative responses, and therefore those cells are growing much faster and they're out competing all the other cells in your iPS population, such that you end up now with a cell type which is altered because it has increases in copy number variation, but it becomes your predominant cell line. Now, the issue here, of course, is that copy number variations themselves are associated with diseases such as cancer. So what we really don't want to be in a situation is using iPS cells that are carrying genetic mutations that have been acquired during the reprogramming process, such that you say, well, I'll create some cells in the laboratory and put them into this patient A because they have a disease and we can cure them with the cellular therapy, only to then find out that cells you've given them are actually cancerous or cause some other genetic mutation, disease, sorry, in that individual. So we really don't want to have that situation. We must really take care of the genetic integrity and the genetic makeup of the cells that we're using when we think about translational therapies. Okay. So what we then need is a cell type that is easy to isolate from as many patients as possible. Ideally, we want this cell to be clonal, highly proliferative, passageable, and bankable. And the reason clonality is an important issue is that if you think about how we get skin fibroblasts, we take a skin punch, and in that skin punch, you're going to have thousands of cells from that individual. When you take that population of cells, it's a polyclonal population, and you try to reprogram that polyclonal population. Well, within that population, only some of those cells are going to start to become iPS. The downside of this is that when you then produce your iPS cell clone, which would have come from just one of those cells, of those thousands of cells you have in the mix from the original biopsy that you took, when you try to look at the genetics of that cell and what changes may have occurred during the process of reprogramming, you have to compare your monoclonal iPS cell to your polyclonal population that it was originally derived from. Now, there's another problem that to do with skin fibroblasts that I haven't mentioned yet, which is that a report that came out a few years back looked at the number of mutations that actually already exist in skin fibroblasts, and they found that within about 1 centimeter of skin, you have around 140 pre-cancer mutations just sitting in those skin fibroblasts, waiting to change and transform. So in other words, skin, because of its locality in the body, because of exposure to UV, etc., these cells are potentially already primed almost to become cancerous if under the right conditions, and therefore putting those cells through a process which might increase their copy number variations anyway and genetic mutations anyway is not necessarily a great place to start. Okay, so we also want the cells to a cell type that can be isolated at clinical grade. We want it to have a genomically normal and stable genome in the first place, and we want it to have a high capacity for reprogramming. The iPS cells themselves that we generate from this original somatic cell that we start with have to have appropriate pluripotency and differentiation capacity, and have a normal genome by the end of the process. So we don't want to do what we saw in the paper I showed you from Nature in 2011, where we're just increasing genetic mutational load within that cell. So what cell types could we use? Well, one cell type that's gained a lot of interest at the moment is urine cells. And it's because they're so easy to obtain. You can literally take a urine sample from somebody, they can pee in a pot, you can take that urine, you can then spin it down, and from the pellet you form, you lay that pellet down in some growth medium, and lo and behold, you can find cells that will grow clonally out of it. And essentially these are thought to be sloughed off kidney glomerular parietal epithelial cells, and they grow clonally, so they seem to match a lot of the easy to get clonal. They seem to match a lot of things that we need. But these cells have now been published for quite a few years now, and yet we haven't yet seen somebody do a high resolution genomic analysis on them. For example, like the one I showed you with the CNV analysis earlier on for skin fibroblasts. Because I haven't seen those myself, I'm still a little bit skeptical about using them until that sort of study is done. But if they turn out they're safe, then that's fantastic. But because I haven't seen anything for quite a long time, I'm just not sure that they are okay, given that they may just be cells which are sloughed off from certain parts of the body. That might indicate they're already going through some sort of transformation or change. So apart from urine, what else could we do? Well, there's blood-derived cells, of course. Even individuals with FOP can tolerate venous blood sampling. So this would really suit practically every patient group or cohort that we have. However, if you think about what happens when you take a blood sample and you think about the cells within there, it's a very heterogeneous population. So, you know, you've got T cells, B cells, you've got macrophages, you've got eosinophils, you've got lots and lots and lots of different cell types that are in there. So it's heterogeneous. And also, each of those populations is polyclonal, a bit like taking a skin biopsy from a patient. You're not just taking one cell and growing it clonally and then use that in your reprogramming. You've got, again, a polyclonal starting population, which is going to make studying the genetics of any mutations that have occurred during the process of reprogramming very difficult to understand. The other thing, of course, is that a lot of myeloid cells, particularly think of B cells and T cells, they undergo genomic rearrangements as part of their normal life cycles. So if we went to those cell types and wanted to use those, we may not have a fully representative genome of that individual anyway, as a consequence of their normal biology. So if you look within blood, there is actually another cell type. Now, it's not myeloid in lineage, and I'll talk about this more in a moment, but it does exist. And this is a cell called the late outgrowth endothelial progenitor cell, but it's also known as blood outgrowth endothelial cells. So it has various names, but for this talk, I will predominantly call it endothelial progenitor cells to keep things simplistic. So what are these cells? Well, as the name suggests, they have an endothelial type morphology. And if we look at the cells growing in culture here at the top, you can see they could grow, they have a cobblestone morphology, and they can indeed actually make networks in culture. They also express markers that are consistent with a phenotype that suggests, or an identity that suggests they are endothelial. So you get them from the peripheral blood mononuclear compartment, the PBMNC compartment of blood, which is where you find all the other blood cell types. But as I said to you, they're not myeloid. Here, if we look at these EPCs here, this is the line here, we're looking at a flow analysis of the markers, and we're looking at different, like CD31 here being an endothelial type marker, at KDR as well. And then we have more monocytic markers here. What you can see is that endothelial progenitor cells look much more similar to pulmonary artery endothelial cells than they do to monocytes. You look at the flows. Okay, so these look like they are endothelial as opposed to any other cell type. Now, to isolate these cells, I mentioned you get them from the peripheral blood mononuclear compartment. We have published a paper in JOV, which is free to access for everybody. And this is the reference in the paper here, where you can go and watch a video of how we actually isolate these cells. So the trick to isolating them is that you need to follow our protocol Exactly. Don't try to change anything you think that might work better. It's one of these protocols that you need to follow precisely. But if you follow it precisely, you have 80% plus chance of isolating endothelial progenitor cells out of your culture. Okay, so what does the process generally look like, simplistically look like? So we take our blood samples, we then put them in a Ficol gradient. We spin them through a Ficol gradient, I should say. And from that, you end up then separating the erythrocytes from the plasma and the buffy coat as well. So the buffy coat, that's the bit we want. That's where the peripheral blood mononuclear cells exist. You just basically take that buffy coat, you'll then layer it down into your dish, and you'll put on medium, which is suitable for growing endothelial cells. And lo and behold, within 7 to 14 days after you've done this process, you'll begin to see endothelial cells growing out in your dish, and those are your endothelial progenitor cells. So these EPCs, these endothelial progenitor cells, are available from peripheral blood, as I've said. And as I've mentioned, you can isolate them with 80 to 90% efficiency from all patient types we've tested so far, including those with bone marrow disorders such as leukemia, and also vascular diseases as well. So one of the reasons we know that these cells aren't from a myeloid lineage is because when we take these cells from patients who have got leukemia, and who carry the Philadelphia chromosome as part of the pathology of why they have leukemia, when we look at the EPCs derived from those patients, they do not carry the Philadelphia chromosomes. That shows us that the EPCs are not derived from the myeloid lineage. We've also taken EPCs out of patients with vascular diseases too, and I'll talk about one of those later on in the talk. But it shows you that even with vascular diseases, you can still get these endothelial progenitor cells out of patients. So these cells are clonal in the way they grow. They literally grow from a single cell into a big colony, and they're highly proliferative, which is part of one of the reasons why they're called progenitor cells, as they're able to proliferate extensively. They're also bankable and also compatible with clinical grade isolation as well. Importantly, they don't carry any permanent genomic rearrangements of the types you might get, for example, in myeloid cells, and they have a relatively normal genomes. So if we were to look here, here's a normal carrier type of the cells here, cytogenetic carrier type. But we've also performed copy number variation analysis on these cells as well, and found that they have a normal representative genome of that individual, and they haven't had any genomic rearrangements as a normal part of their biology. So they're a good starting cell in terms of genetics, particularly because of clonality as well. So the basic process we originally started off with, which is how it was done traditionally, because we did this work back in 2012, was to take our endothelial progenitor cells. We then use retroviral delivery of Yamanaka 4 factors, and we put them onto our cells. And either we used 100,000 cells in one well of a 6-well plate, or we tried putting 4,000 cells into a single well of a 96-well plate. So when we originally did the experiment, we used EPCs from healthy individuals and also patients with endothelial cell defects to show that even from patients with endothelial cell defects, we could isolate the EPCs, but also reprogram them as well. And as I've mentioned, we use retroviral expression vectors. So at the end of the reprogramming, we find that they've expressed normal pluripotency markers, as one would hope they would. And also they're able to differentiate in vivo into all 3 germ layers and derivatives of those. So they appear to be pluripotent and grow normally. But what we didn't expect to find, which was quite interesting and again could be very useful for the translation of this work to the clinic, is that here, if we look at the efficiency, at the top here, we're looking at blood-derived cells. So in other words, blood-derived endothelial progenitor cells versus skin fibroblasts at the bottom here. So all of these represent skin. The top bits are the EPCs. In each well is one well of a 6-well plate here. And in this, we're looking at one well here of a 96-well plate. So here we put 100,000 cells, and here we put 4,000 cells into each well. The black dots you can see represent alkaline phosphatase positive colonies, which in other words represent our iPS cell colonies growing out. The first thing you can see is that looking in our 6-well plates, The timing is such that you're seeing iPS cell colonies emerge very quickly using endothelial progenitor cells. So by day 10, you can already see alkaline phosphatase positive colonies. And over time, those colonies grow very rapidly and are very robust. And we have lots of them. When we look at the skin, we get fewer growing relatively, and they take longer. And importantly, you see a lot of partially reprogrammed cells filling up your dish. So this messy kind of grainy look to this is because they are all these partially reprogrammed cells. We don't really see that here with the blood outgrowth cells. And the reason we think this is happening is because when you take endothelial progenitor cells, they only grow well in endothelial growth medium. When you switch them into medium that contains DMEM, which is something we find in most reprogramming media, they just die. They don't like that. So in effect, what you're doing is you're wiping out all the cells, all the EPCs that are not reprogramming, and all the EPCs that might only be partially reprogramming, leaving you only with the cells which are fully reprogramming. And that's why we get nice, really clean colonies from here. Now, cost efficiency looked really high. So we said, well, let's do the same experiment this time and see if we can, if it would fit with some sort of high throughput method. So we took only 4,000 of the cells, as I mentioned, and put them into one single well of a 96 well plate. And we did obviously 12 replicates. These rows are 12 replicates. What we ended up finding was that with EPCs, you found between 1 to 3 colonies per well, whereas if you looked in skin fibroblasts, you didn't see any with just 4,000 starting cells. So the efficiency is quite high. And if one wants to put this into a 96 well format, it would probably work well. So what about the genomic stability? Because we already heard that skin fibroblasts are really not good in terms of their genetic integrity, partly because maybe it's a polyclonal population, but partly because they may be picking up mutations during the process of reprogramming. So we did the experiment where we slightly differently from what you saw earlier on, because instead of using a SNP array, which is how they looked for the copy number variations in that Nature paper I showed you earlier, here we used array CGH analysis. In this analysis, what we found, looking at copy number variations, here we have the reference genome, which is our EPC line. So patient 1, patient 2, and patient 3. I apologize for these lines, they've shifted. They should be further down. And then the iPS cells derived from them. What we found is of the 11 lines we tested, 9 of them showed no copy number variation at all. So, you know, compare that to the hundreds of copy number variation we saw early passage at a similar passage, which is what we did here. We did passage 5. You can see that instead of hundreds of copy number variations, we're seeing no copy number variations. And those that did get copy number variations were only very few. So this is obviously unsurprising and interesting to us. And one question that somebody might ask is, well, why? Why is it that these genetics look so good? And well, one is because they're clonal, I think, making our comparison between starting cell and iPS cell line is much easier because our original cell line was clonal. So our reference genome for doing this analysis of our somatic starting cell is clonal and our iPS cell is clonal. So you're really doing a side-by-side comparator. So on the one hand, it looks like the genetics are good because of clonality. The other thing that might make EPCs better at reprogramming is that if you look in their genomes, not genomes, their expression profile, you actually find them expressing NANOG and REX1 already. So they already have some sort of pluripotency network markers already existing in them, which may mean it makes it easier for them to move state or to shift identity into an induced pluripotent stem cell state. So this again might make the reprogramming process less stressful for the cells in themselves. Whatever the reasons are, ultimately, what's critical is that we're seeing less copy number variations. And even if it turns out that it's only because we're doing monoclonal population versus monoclonal population in our comparator, in our ACGH, the point is that if you want to study the genetics of your iPS cells and how they might change or what mutations they have acquired, using EPCs seems to be an advancement that's very helpful for that. Okay, so I mentioned to you that we also looked as a proof of concept, proof of principle, said, well, let's look at diseases which have endothelial cell defects in them. And the disease we were looking at here was pulmonary arterial hypertension. And in pulmonary arterial hypertension, you actually have endothelial cells which actually become much more proliferative. So one of the reasons they become more proliferative is because they actually have abnormalities in their chromosomes. Basically, they get genetic mutations and copy number variations in them. So using that cell type, we decided to say, well, when we do make our iPS cells from those, are they going to become more unstable and generate more copy number mutations as a consequence of being reprogrammed? The short answer is no, they don't. So you can still make iPS cells from them, and they don't gain any more copy number variations in the process of reprogramming. So, of course, when you're studying diseases of this type, where you already have some sort of genetic abnormality or copy number variations, this is essentially a game changer, because you're not creating any more mutations in your reprogramming process. So then when you get your iPS and then you go forward and differentiate those, it will make your understanding what is going on in your cells and what new mutations might be acquired as they become a certain cell type, or how you treat them with certain chemicals or whatever stimuli you need to push the cells into more of a disease phenotype. These cells can really help you answer that question. Okay, so the next part of the talk is I want to think about, so that was our initial work in the identification of EPCs as a reprogramming substrate that we could potentially use. How do we now take that forward in a clinical setting? So there were some disadvantages as well as advantages of what we did before. So the advantages, just to summarize, were that these cells, EPCs, are clonal. They are easy to obtain from blood from virtually every patient group, and they have a genome that's representative of the individual they come from. They show good reprogramming efficiency, very high reprogramming efficiency, in fact, and the iPS cells that we derive from those EPCs appear not to acquire any new copy number variations or very low numbers of copy number variations compared to their starting cell. But the caveats, of course, are that at the time, we're using about 60 mils of blood in order to isolate our EPCs. We were also relying on fetal bovine serum. Of course, this isn't xeno-free and is incompatible, generally speaking, with clinical trials, although you can still use things if you can trace things properly, but ideally we want things to be xeno-free. We also use retroviruses, which again are not things we want to use clinically, and we also used mouse embryonic fibroblasts as our feeder cells for these cells during reprogramming. And again, this is not xeno-free, so we need to at least overcome all of those things. So the first thing we did was try to improve EPC isolation by reducing the volume of blood. We tried a whole series of different compounds and chemicals and other things, and we actually found the best thing, the thing that most dramatic change, was switching from using fetal bovine serum to using commercially bought human AB serum or an autologous serum from the patient themselves. The reason we were using fetal bovine serum, we had to use a very special kind of fetal bovine serum, which was, it had to be ESL qualified. That was an important part. If we just used normal fetal calf serum, it didn't work. So we had to use ESL qualified FBS. Here, of course, this isn't the serum we're using isn't ESL qualified. It is just normal commercial AB serum, but it seemed to make the biggest difference. Now I'm going to show you a comparison now of the things I'm talking about. So here, if we look at ESL qualified bovine serum, and this is commercially bought a human AB serum. If we start off with 40 mils of blood from an individual, we will get about 1 to 2 colonies of EPCs using this ESL qualified fetal bovine serum. But with the commercially bought HB serum, this dramatically rose between 4 to 6 times higher by using the AB serum. We suddenly got 4 to 13 colonies per culture. So based on that, we got quite excited and thought, wow, actually, what if we now reduced from 40 mils down to 10 mils? Because ideally 10 mils now, this is something we could now go to children to very easily, because one of the things that's stopping us from being able to use EPCs from all individuals was, well, 60 mils. It's really not that much, but some people, some pediatricians are like, well, 60 mils is a bit much for this kid, or I don't really want you to take that much. So we said, if we bring it down to 10 mils, what do you think of that? And said, yeah, 10 mils is fine. So we did that. We took it down to 10 mils. And because we had such a high level of colony formation using human serum, when we took it down to 10 mils, we still were in the right ratio. We got 1 to 4 colonies of EPCs growing out of 10 mils of blood from individuals, and we did this on children and adults, and it works very well. Like I said, 80% plus of the time we can get colonies growing out from these. The ESL qualified fetal bovine serum that we use, of course, produced no colonies because we're only getting 1 to 2 from 40 mils. So bringing it down to 10 mils was no surprise. We didn't see any colonies. We also tried using autologous human serum. So in the process of isolating EPCs, as I mentioned to you, we spin the blood through a Ficol gradient and we end up with our buffy coat, and above that we actually have the serum. So we just collected that serum without treating it, doing anything special to it. We literally just took it and we used that as our serum that we used, and that again produced 1 to 2 colonies out of 10 mils of blood. So this seems like it's really a useful way. If we can use human serum, we're xeno-free, and that also increases the efficiency of isolating EPCs quite substantially. When we went down to 5 mils, though, we really began to struggle. There was certainly a cutoff around 10 mils, but after that became more difficult. So although 1 out of 6 of our patients we could isolate EPCs from, it's not something we would recommend routinely. 10 mils seems to be the limit. Okay, so the next thing then is to think about the delivery of the reprogramming factors. We'd used retroviruses, as I mentioned to you before, very high efficiency, quite helpful. We tried many different things in the interim, including piggyback, sendai, episomal, but they all had low efficiencies of reprogramming. So we then next tried messenger RNA. And in fact, what we found was that they reprogrammed really well with messenger RNA. So here I'm showing you non-modified messenger RNA, and the RNA that I'm going to mention here now, whether it be non-modified messenger RNA, and I'm going to talk in a moment about self-replicating RNA, all these are available from REPROCELL. If you contact them, some of them are available as kits, some of them not, but if you speak to them, they could even do custom orders for you where they can make up the RNA and produce it for you. So I'd speak to them about that. But essentially, let's start with non-modified RNA then. So here you take your EPCs, you lay them down, 100,000 cells, you do a series of transfections over 8 days, so 8 transfections in total of this non-modified RNA. And this is basically a cocktail of 4 individual RNAs of reprogramming factors. And then by day 14, you can begin to see colonies forming on your dish, which you can then begin to pick. And here I'm just showing you the time course using TRA 160 Stain Alive, again available from REPROCELL. And you can see that the cells light up with the staining for these TRA 160, and these are live cells. So it's really helpful for identifying colonies that are reprogramming correctly for you to pick. If you look at the cells you pick then and you stain them for the normal pluripotency markers, you can see they express what you expect them to express normally, which is great. And doing a normal cytogenetic karyotype, they look normal as well. And these cells also can differentiate in vitro and in vivo into 3 germ layers and derivatives thereof. So non-modified RNA is certainly usable on endothelial progenitor cells, and it has a good efficiency of reprogramming. An alternative to that is to use self-replicating RNA. The reason for using this is that some people, especially when you're trying to make a process to generate iPSLs from lots and lots of individuals, doing those 8 transfections can sometimes be a bit time-consuming or laborious. So an alternative way is to use this Venezuelan equine encephalitis virus, which has been published in Cell Stem Cell in 2013, to use this as a way of having an RNA within a cell that you only need to transfect once, and then it will remain in the cells and constantly produce your reprogramming factors and reprogram your cells over time. And then obviously over time, it will dilute and disappear eventually and be lost from the cells. So this is certainly an advantage when it comes to reprogramming if you want to spend less time doing transfections The downside of it, though, compared to the non-modified RNA, is it takes slightly longer to produce colonies. So we get them by day 20. You can pick, as I mentioned to you before, from around day 14, 15 is when we were picking with a non-modified RNA. But again, I said, handling wise, it's much more simple. You literally do one transfection of microRNA, one transfection of the self-replicating RNA, and that's all you have to do. The other thing that's in this medium that's slightly different from the other one is that we use B18R. B18R is a protein that's used to dull down and dampen the interferon response so that cells don't reject the RNA that's in them, by thinking it's viral RNA, they need to attack it and destroy it. You're just dampening down the interferon response, so this self-replicating RNA can be maintained in these cells. Okay, so here again, a time course like I showed you for the non-modified RNA. We have the so you can see by day 21, you see some nice colonies that are easy to pick. And again, we really found this trial 160 staying alive really helpful for this kind of thing, because we don't have to kill the cells, fix them, we just put it on alive. And so we can just wash it off and it's gone later, and we can still use the colonies for what we want. Pluripotency markers are still all expressed as we would expect as well, and these cells can differentiate in vitro and in vivo, as I mentioned with the other cells, the other non-modified RNA. We also can get differentiation into the 3 germ layers and derivatives thereof. So both types of RNA, whether it be non-modified single messenger RNAs cocktails or the self-replicating RNA, both work well in reprogramming in our iPS cells, in our EPC, sorry, to produce iPS cells. The next thing we considered was matrix, because of course we mentioned to you before that we're using mouse embryonic fibroblasts. So again, we tried lots of different things, not all mentioned here. We tried collagen, fibronectin, vitronectin, matrigel. What we found actually was something called laminin 511 was really, really good at improving the efficiency of reprogramming. You can see the numbers here relatively next to each other. And this is xeno-free and obtainable. It's human laminin 511, commercially available. And again, I would recommend you can use that instead of other matrices that are currently being used, because it has a very beneficial effect on reprogramming. So what about efficiency of reprogramming then? Well, here I'm showing you the data from the self-replicating RNA. And just to add in here that the non-modified RNA actually has a much higher rate of, or much higher efficiency of reprogramming. But I'm showing you, as it were, worst case scenario here for RNA. So with the peripheral blood here, you can see you're still seeing 0.025. And although this, you might think, oh, that doesn't sound like it's very high compared to, for example, the retrovirus gene delivery we mentioned. It's true, it's not as high, but the quality of each iPS cell clone is a lot less variable. And most importantly, they show a very high level of complete reprogramming. So we have found in our hands that take 100,000 cells, you get at least 3 colonies per well from that. Each of those colonies that you pick, every single one has turned out to be usable as an iPS cell line. So in terms of numbers, what's more important, it's not how many colonies you produce, it's how useful they are to you at the end. This method really appears to give you usable iPS cell colonies at the end of this. There are no obvious partially reprogrammed cells, as I mentioned when we use retroviruses too. And again, this of course then reduces the time taken to obtain a clonal iPS cell line for downstream use. So that's really beneficial as well. Okay. Okay, so if I were to now look at, instead of looking at one individual, but look at whole groups of individuals, if I were to take here, we're looking, 60 plus individuals, we've got EPC lines from, how many of those will reprogram on the first run? We found that around 80%, whether we use fresh or frozen blood EPCs, we can reprogram them first time for about 80% of people. Those that didn't work on the first run, on the second run, they did work. So we should, in theory, be able to produce iPS cells from EPCs for almost any individual that we can get EPCs from, okay? And that the EPCs, as I mentioned to you before, we get them from around 80 to 90% of individuals we can get EPCs from. So, you know, hopefully this is something that will work for the majority of individuals. Just to show you about, we talked about the self-replicating RNA and retention. We're just showing you here that by passage 3 to 4, your self-replicating RNA is undetectable, so it does disappear. Partly because I think you're removing the B18R, so therefore that RNA is now not, the interferon response is not being dampened down. The RNA can now be attacked and destroyed over time, which I think it is. When you look at the carrier typing of the self-replicating RNA produced iPS cells, they also have a normal carrier type, just like the non-modified RNA. But obviously what's happening at the higher level resolution. So what I just showed you was cytogenetic analysis, and in that sort of analysis, you can't go lower than about 5 megabases. So this time, instead of doing array CGH analysis, as we did in our first, the first copy number variation analysis I showed you earlier on for EPCs, here I'm using SNP array analysis, just like they used in the paper, in the Nature paper in 2011. So we wanted to make it as close as possible, use exactly the same system as them to see how many copy number variations would we get. So the first thing to say is that here, if I just now look at EPCs reprogrammed by self-replicating RNA, or I look at fibroblasts reprogrammed by self-replicating RNA, we see that we see fewer copy number variations in our individuals, between 1 and here 3. But interestingly, even the fibroblasts, where we still would have expected to see hundreds, 300 or so, up to 300 copy number variations, with the RNA, you see less copy number variations being accumulated. And this could simply be because this is a non-integrative method of reprogramming, again, that's beneficial even for fibroblasts. If you want to reprogram them with RNA, it looks like this is still a better method to use. But now let's look more about what happens with self-replicating RNA in EPCs, which are programmed by self-replicating RNA or by retroviruses. And what we find here is that we see, again, very few or no copy number variations when we use self-replicating RNA. Whereas with retrovirus, we still see copy number variations, although low copy number variations, as we already saw before with array CGH analysis, although not 0, we see low levels. Again, nothing like the hundreds that we saw happening with skin fibroblasts. And here again, here patient 6, you can do side-by-side comparator, you see a much more reduced number of copy number variations. So overall, if we focus on this entire column here, you can see of all the lines we analyzed, only 2 out of these 8 actually showed some copy number variation, and it was only 1 and a very small region as well that was being lost. So overall, I don't think we're ever going to find a method of reprogramming that's going to be totally free of causing some sort of copy number variation. And some of these might just simply be cells just proliferating normally, causing copy number variations in the population somewhere. But the vast majority of lines, at least, there's a higher chance of them coming out being genetically unaltered, which were certainly at the copy number variation level, which would allow translation of these cells into the clinic more readily. So I think that's a helpful step. So finally then, we said, well, let's do the whole process clinical grade. In other words, let's get EPCs, use all xeno-free conditions, human serum to isolate the EPCs, then let's reprogram them with RNA under, again, xeno-free conditions, all which would be compatible with clinical translation. And the cells we produce are the same as I've shown you previously. They express the right pluripotency markers. They can differentiate into all 3 germ layers. So it looks like this method could be taken forward into translational applications. And just to mention that REPROCELL and Roslin CT are actually working currently on trying to make clinical grade cells for use for the community. And that's very interesting because obviously success of this project would be great because we could think about now creating hematopoietic stem cells from these cells to treat leukemia in the ways I've mentioned earlier on. So for example, we know that we could take an individual who has leukemia, we could generate EPCs from those and such that they wouldn't carry the mutations because they're not myeloid in origin from that individual that's causing their leukemia. We could then create their iPS cells and then with the right differentiation protocols, make them into hematopoietic stem cells and then replace those for individuals on an autologous manner. And the other way, of course, is thing we could do is make endothelialized stents. So because my interest of my group is in cardiovascular diseases, we make smooth muscle cells, endothelial cells, cardiomyocytes, etc. And there are good, robust protocols for making endothelial cells now, but enough of them that we can actually endothelialize a stent. Which you can't do with endothelial progenitor cells because there aren't enough of them. And more importantly, endothelial cells are not all the same, just to be clear. Just because something's called an endothelial cell, it could be arterial, it could be venous, it could be macrovascular or microvascular. They're not all the same. So you want to make exactly the right type to go in your stent. And that's something we're working on, which is why it's interesting to us. But if you could do that, those stents then would not be rejected so easily. They wouldn't calcify as easily. And that could make, again, a bit of a game changer for treating coronary disease and other sorts of conditions where you need stents to be put in. So then to summarize, I've shown you that there are several hurdles we have to overcome. Cell substrate and reprogramming methods are key factors we should consider. I've shown you hopefully that combining EPCs with RNA reprogramming has a number of advantages. And from this work, we could now start thinking about translation potentially, and work is going into that. And I want to give a little call out here to REPROCELL's GMP IPS master cell bank manufacturing service. And I'll put a slide of this on in just a moment. So they're working on this and it's worth you having a look at that if you're interested in having some cells made for you. But before I show you that, just wanted to acknowledge the help of my group and my collaborators in Cambridge when I was there, who helped me with the publications of the work that you've just seen. So in particular, Professor Nick Morell's group, Ludovic Vallier's group, and Alan Bradley's group. And then a big shout out here to Sarah Eminley, who has been fantastic in helping us from REPROCELL and did a lot of the work that you can see on the RNA reprogramming. So a big call out to her and a thanks to her for all her hard work in getting that to really bring that all to fruition. So here now is the REPROCELL's GMP IPSC master cell bank manufacturing service and information on it, which you can get more information from the website as well.